So what I was asked to talk about was just to bring you a little bit of a presentation of some of the work we've done. It has a lot of the stuff that I've done, but a lot of stuff that comes from other groups, but a lot of very big influence coming from the MMRF for sure. Particularly the majority of the work I've done the last probably 7 or 8 years has largely been exclusively oriented with MMRF. So I've been very fortunate to be involved in some really nice studies, actually. So one thing, you've kind of seen it from Louise, and we've started talking about it, is this idea that people will say that you have multiple myeloma. And probably everyone here, when someone asks you, you say, I have multiple myeloma. Now, the problem is that, is there a pointer? Sorry. So this is just basically because the pathologist looked at something, and this would be a normal bone marrow, and here's a myeloma marrow. And they, based on looking at this under a microscope, go, you have something, because it looks like something, that we call multiple myeloma. Now, the problem with that is, at least from the scientific side, and maybe some of the clinicians will disagree at some level, when we go and zoom in on each one of these tumors, and we can pick 1,000 different technologies, or probably really more, 3 or 4, but we pick a different set of technologies, we can actually start looking at patients. So here, each one of these lines up and down is an individual patient. And what the important thing here for you to see is there's these little boxes here that go down. You can kind of see each patient kind of separates into different groups. And that's really this, to try and illustrate to you that myeloma is not one disease. We call it multiple myeloma, but it is kind of the frightening side from my perspective. We say it's myeloma, but really you should be, I have this type of myeloma, or I have that type of myeloma, I have that type of myeloma. The good caveat being that is that some of the therapies that are available right now have been working so well, they seem to work across most of the subtypes. And where we probably really need to focus now is understanding which of these little groups are not responding to the current therapies is probably, for immediate return to patients, what we really need to understand. And that's one of the things that will come out of COMPASS for sure. So if we jump back, and my other thing is history. So if you were to walk all the way back to the 1980s and say, what did we know about multiple myeloma, at least from genetics, omics? Well, we knew there were 2 myelomas. And again, this is back to that idea, there's different types of myeloma. We could separate patients just looking at their chromosomes. So if we take your cells and we pop them on a slide and we look and see how many chromosomes are in each tumor cell, we could separate them into 2 broad groups. One we call non-hyperdiploid, so they basically are more or less normal. And then hyperdiploid, which is this kind of weird thing that no geneticist really understands, which we say, for some reason, the odd number chromosomes, so chromosome 3, 5, 7, 9, 11, 15, 17, 19, will have 3 copies instead of the normal 2, which doesn't make a lot of sense. And then to really make things complicated, if you like looking at other related diseases, so the acute lymphocytic leukemias, they actually have the exact opposite phenotype. They have hyperdiploid, but then all the even number chromosomes. And it's kind of weird how whatever the disease entity of multiple myeloma knows to copy the same odd number chromosomes, and a different disease entity knows to copy the even number chromosomes. The sad part is this is about half the patient population. We don't really understand what causes that, at least at the genetic level. These guys over here, we generally have a pretty good understanding of what causes that that subset of myeloma. Then we can move into the 1990s, and we got a little bit smarter. We could actually paint each chromosome, and you could start getting a better understanding of how complex things are. So instead of just those gray bars, now you start seeing every chromosome had its own color, and then you get over here and you go, oops, there's a little bit of this chromosome over on this one. There's a little bit of this chromosome from down here, up here. We could start getting an appreciation for how complex things really were. And then we move in, and this is my personal plug because this is really what some of the stuff I did, and we'll make sure we go. So this is what, using a technology we call comparative genomic hybridization, or CGH. And again, the technologies aren't important, but really just the picture's worth 1,000 words. This is quote unquote normal if you consider males normal compared to a female. This is a myeloma patient compared to an average female. And you get to start to seeing how different the tumors are changing. So it is a fairly difficult landscape that we're dealing with, all the changes that can happen, and trying to zoom in on all those changes and figure out which one of those things are actually important and which ones of them are not. The good news is we're getting very close to being able to do that. And a lot of that has to do with what we've been doing now for the Multiple Myeloma Genomics Initiative, and now following on with COMPASS, where technology has really moved to the point where we no longer are limited by the fact of going, I wonder what's going on. We can always figure out what's going on, and pretty much there's an issue of funds, but for any one tumor, we can pretty much tell you what is wrong in that tumor compared to normal today. There's very limited that we can't. And what I'm going to use here is my little class project. So everyone in here we'll consider is one of the genes in the genome. And we generally associate cancer as being a genetic disease. So anyone who got one of my cards, please stand up. And I have no idea which cards I gave to people. So the important part here is we just sequenced this myeloma tumor genome. And each person who stood up is a person one of the mutated genes in the genome. But the problem is there's only one of you that's actually driving the tumor. So the first thing, and we'll go into this in another one of the next slides, is we can do some things to try and get rid of half of you. And that's basically going to be which one of you is expressed, can talk, respond. So anyone who's got a card that has a square on the back, you can remain standing and the rest of you can sit down. Okay, so we've basically been able to quickly go through and say, okay, these ones were just what we call passenger mutations. You guys got to brought along for the fun ride. But some of these guys here are important. One of these guys is important. The rest of them, again, are just passengers like all the other people who sat down. So who's the lucky person who's got the card on the back that says troublemaker? So she's our problem. So that's the person we need the drug for. Now the problem, what I could tell you is, what we can do today really well is I can get the first group of people to sit down. I can't tell you for sure how to make the last batch of people sit down so that I say, now to Onyx, Millennium, and every other 30 pharmaceutical company, I need something that gets rid of her. I want to make her sit down. You can sit down now. Thank you very much. But that's really where we're at. So we've gone so far. We've been able to go down to, we can tell you which genes are the problems. We can get rid of them, about half of them. Now weeding down to the last bit is the hard part. The way we're going to get over that is when some statistician comes in and says, well, if you just look at enough people, you'll figure out which ones are common, and those are the ones that are the troublemaker. And that's really why we wanted to do 250. But that was back in the day when we thought myeloma was myeloma. Now that we look at different subsets to be able to know what are the important things in each subset, that's why we really do have to expand out to a bigger project to get the numbers to understand which things are important in each individual subset. So the way we do that is now with what they call next-generation sequencing. And this is just an example of some of the sequencing data from some myeloma patients, where we can see this is the DNA level, and we can pick up where you see this color area is where there's a mutation in the DNA. And then we also, if anyone knows, there's something called the central dogma of biology, which is you have DNA in your cells. That DNA gets turned into RNA, which is called messenger. It goes out and gets turned into the protein. The protein's really the workers. And at the end of the day, if you don't get turned into protein, which is the first group of people that sat down because they don't make that RNA, the protein is where the problem is. That's the real working body that we need to figure out. So here we were able to look at this sample and go, okay, there's a gene that's mutated, so all the people who stood up, and it's actually also expressed. So those are the people that stayed standing in the second batch. But what we can also do, and we do this with Compass now, we can see here's another gene that was mutated, but the gene's not expressed at all, so it's not doing anything in this tumor cell. It just came along for the ride. So this is one of our easiest ways where we can, again, separate a large portion of the population and split them down. Why we're doing not just 1 or 2 assays, we're doing about 3 different assays in Compass. And why do we do all that is because it can make a pretty picture like this, in case you care a pretty picture. And this is just a simple, a very complex diagram, but a way to represent, depending on time frame, but when this sample was generated, this is probably, you're looking at about $100,000 worth of data smashed into one picture. But basically, what it's showing you is all the times where that copy number has changed. So you remember that slide where things normal to tumor, how it was jumping back and forth. All the genes that are mutated, and we've color-coded them, so the ones that are in red are actually the important genes. So you can see how many of the genes along here get mutated but really are not important. And this is where it's really hard and why it's important for us from the scientific side to figure out, separate these things that are just coming along for the ride versus things that are really there and really important. And then also, if you think of that other colored diagram where you saw different chromosomes coming together, all the lines to the center here are marking those kinds of events. So we've been able to actually bring all that information that you saw kind of slowly accumulate over about 30 years. We can do that now in about 1 to 2 weeks and then make a very simple representation of the overall problem. Now that's kind of what we can do, again, for the idea of individual myelomas and separating different patients. But one of the things that's really important that we've started to realize is not only are each individual patient different, but within the patient, there can be differences within the patient themselves. Now, they're not as broad, but they can have probably even more importance for therapeutic outcome. And a simple way of looking at this, and this is just one of my kind of teaching slides that we use frequently, is to break this down to a military comparison. So this is, at the end of the day, myeloma is related to your part of your immune system. Your immune system is really our army out there fighting off infections. So here's the draft, and these guys get pulled into the draft, and they go all the way through their physicals and make sure they're ready to go out, and they get finally approved. They go off to basic training. They go through their basic training, and at the end of the basic training, they get a furlough to get out and go have some fun. And one of these guys has a little bit too much to drink one day, picks up something he shouldn't have picked up, or she, and decides to go AWOL. And that's where that myeloma cell comes from, is that guy that went out and had a little bit too much to drink and went AWOL that next night. And the problem is once he goes AWOL, every time he goes and takes another step or divides, another cell comes out. And sometimes each time they're subtly different. And every once in a while, one of those ones that comes out has a huge advantage and explodes and takes over the population. But that kind of keeps happening burst by burst by burst. We get to another phase where, again, they're spitting off other types or other kind of distant relatives, and then one takes off, and that's really where we might find the diagnosis. Now, this is where, again, the next part gets a little bit more tricky from the study side, is understanding once we know that we have this big bulk of a tumor, it again is spitting off different ones. How does therapy affect what happens next? Does therapy cause the same tumor to be there at relapse? Does one of the new derivatives come back? Or does it actually get rid of all these things and maybe we go back to something a little older? And those are things that we started to really look at recently. And we do a very simple thing. You look at the tumor when it started, and we look at it at the end, and we just ask, what do they look like? Do they look different? Do they look the same? And you can take a simple model that at diagnosis, say this tumor is all the red dots, they all look very similar, versus this tumor is maybe a mixture of 2 different dot types. When we treat a patient, what happens? Maybe one treatment kills off all the red dots get killed off by the treatment, but doesn't really affect the other one? And this is one that's really important and personal from my side is a lot of our drugs give you partial responses, or maybe you get into VGPR. The question we don't know clinically or scientifically is, did we just knock out one group of the cells but leave the other ones? And wouldn't you love to know that, you know what, you had a therapy that was 100% effective for 80% of the tumor. Now let's please stop using that therapy because it's not working on that 20%, and let's change. We need to find ways to be more nimble to do those kinds of things. I don't think I would ever propose that we're there yet, but in the future, I think those are the kind of things that we need to be doing for sure. And we can just ask that simple question. If we look at diagnosis, did we just debulk it a little bit, or does it change? Does it look different when it comes back? So we did this as a study that we spent an ungodly amount of time working to get published for something that seemed very basic. It took a while to get out. But we just asked a very simple question. If you look at a tumor at diagnosis, and then we look at it at relapse, how does it look? Does it look the same? Does it look different? The textbooks say it looks like this at diagnosis, and it keeps moving down the line. Nice linear train. This is why your physician will never give you the drug that you got at diagnosis at your 3rd relapse, because that didn't work after the 1st relapse, so why would I go back to it? And that's just the dogma. Everything's linear. And that's probably wrong. So at diagnosis, what we saw is about 35% of patients, they look the same. They didn't change. Now, this goes kind of against if you read the first paragraph of almost every myeloma scientific paper out there, you'll hear them say multiple myeloma is a highly unstable genetic disease or a highly unstable B-cell disease. A lot of patients aren't changing much with time. And those are probably people that really would benefit from, they got really nice responses, and maybe their tumor didn't dramatically change. We should be able to go back to some of the same drugs. Then there's a very small percentage of patients, about 20%, that really follow that classic model. The only thing they got, the only changes were just new changes to what was already there. They just kind of continued along that linear trend. The other group, which is probably the majority, are a little harder to understand. And it's a little, this where we're looking at the diagram will help. So in this diagnosis, they have a red bar, a blue bar, and a green bar. Those are the different genes that are mutated or abnormalities. But there was an earlier cell that had the red and blue, but didn't have the green. And what happens is that relapse, what we realized down here, is that green wasn't actually there. And we can't actually, unless we're the hand of God, I can't go in and make something reappear or disappear that was there once. So actually therapy got rid of this guy. But what came back was not his downstream or his offspring. It was way back, it was someone that came from grandma or grandpa. And this is a little bit kind of what we're starting to call the branching evolution of cancer, that sometimes what we see is one arm, and then therapy is really selecting that arm away or getting rid of it. But what comes back is something different. And one way to illustrate that point is one of the patients that was in this study, we actually were able to look at over 7 time points across the entire disease course. And we were able to kind of see how things changed from diagnosis to relapse 1, relapse 2, relapse 3, how different drugs, carfilzomib was in there, bortezomib was in there, lenalidomide was in there, and how those things modified what the tumor looked like. And this is kind of the story of the cat came back. If you remember that little cartoon that was used to be on, well, in Canada it was Superstation, I'm not sure what it was in the US. But diagnosis, this little green area here is just a region of the genome that's missing in the tumor at diagnosis. And what you see is at the first relapse, that region goes back to being normal, where everything's all black. But down here, something new appeared. And then after that patient received, so this was lenalidomide treatment. So lenalidomide killed this, but this thing emerged. Then this person got carfilzomib and then followed on by bortezomib. That killed off this guy, and this one came back again. And then they went on to MPV, and that actually got rid of that, but this one came back again, and this is really the bad actor that eventually was the one that caused this patient to succumb to the disease. So we're not always just fighting that person's tumor. Sometimes there's 2 tumors within each person, or maybe 3 or 4. I don't want to say there's thousands, because I don't really necessarily believe that's the case, but probably 2 or 3. Some patients will have 2 or 3 different tumors, and it does have to be where we have to be careful. Again, that idea, if you get maybe a partial response, does that mean you got rid of this guy, but then this guy is still sitting there because it was 10% of the tumor to start with? And where that's important, and this is just a cartoon, you can actually see we actually mark the different subtypes. And just in this pie chart, you can kind of see how this tumor evolved and ebbed and flowed with time as each of the different ones kind of came and went. And then all of a sudden, this very bad actor emerged at the end. But the important thing here, and why I was so happy to get involved in Compass, is what this did for us is we could track all the changes at each different time point. And at the end of the day, what we really want for therapy is we want a therapy that would hit all of these different colors. And the way we can get to that is getting to the meat of the problem, that original bad actor that really was the start of the problem. And the nice thing when we look at all these different samples is we can start to see the things that are similar to each one, and you can start seeing where that common progenitor is. The thing that was one of those early events that really is common to all the things that sprout out of this problem child. And at this point, in this patient, was at any given time point was pretty complex, verging on about 50 changes at any given time point. But actually, if you go back and you look at all the different time points and you figure out what's the one things that are common between all those time points, there's only 5 things. Now, if I could make half those 5 things sit down like we did, well, there's probably drug companies in here. If I gave them, they got a 1 out of 3 shot, there's probably drug companies that'd be interested in taking a 1 out of 3 shot. And that's the kind of power that we're going to get from doing this serial stuff with Compass, is being able to really narrow down what's the things that are really important for us to go after in the future. And then I have my pretty ending slide. Thank you.